Our major aim was to do the training at the master's and the PhD level, because in Africa we have very, very few rodent scientists who are scattered. So for now, we managed to do the training at the PhD. We did training to East Africa. We have students from Ethiopia, students from Uganda, students from Southern Sudan, students from Rwanda, Malawi, Zambia, and Namibia.
My MSc first was registered under the Department of Wildlife Management, and the master's was Masters in Wildlife Management and Conservation. It was all about assessing the effectiveness of the fertility hormones, which are levonorgestrel and cholesterol, on the reproductive parameters of rats. First, these hormones, they are used by human females. So those hormones, they have impact on rats. So we wanted to see the effectability on Tanzanian rats. The main goal is to reduce the young that will be produced by rats.
My research topic was focused on binomic host plasticity and the effect of host-seeking behavior, mating, and the feeding success of predominant bedbug species found in Tanzania. The end of this result of these findings, it provides the baseline information based on behavior of bedbug. So this information will help us later on to know how we can generate the strategies control for bedbug. Because after knowing the behavior of bedbug, how they mate, how they feed, it can help us to conduct or to generate some control strategies.
I'm currently looking at rodents' movement patterns and relating how did this movement associate with changes in seasons and habitats, but also as well as how do they involve in transmission of zoonotic diseases from rodents to humans. For the case of Kilombero, we aim to see, do they really move within that exact range? Or do they move way more than what is written? So what we'll get is really going to be useful for both the government and researchers all over to understand that if these rodents are moving out of their expected or their known home range, it's really going to be easy for them to introduce control measures or to establish control measures that will fall within that expected. So you know you are controlling, but you know exactly where are you going to do the control.
I did my research on the African giant poached rat. These are well known now as hero rats because they are trained to sniff landmine tuberculosis, and currently they are used to detect smuggled wildlife materials in airports. And we have other research which are ongoing, like salmonellosis, brucellosis. They are trained to sniff those diseases. I wanted to know the reproductive biology of these animals, because you can imagine the potential of these animals. According to APOPO annual report 2021, these animals have helped to clear about 70,000 land freed from the terror of landmines. So I wanted to speculate and actually to research on this to know whether these animals have got a specific time where they can reproduce, or they just reproduce throughout the year. Because unless you know this, you cannot maximize the reproduction.
My research is aimed at identifying species diversity, abundance, and parasites in rodents and shrews in Mwanza region. I choose Ilimela district because there is a lot of activities which can bring people there. So they used to interact with rodents and also livestock. Because of the lake, I choose my study to be along the lake, Victoria. I'm looking into the species diversity and then the abundance and diversity of rodents and their parasites. Because we know rodents and shrews, they are vectors of many zoonotic diseases. So since I got those parasites who are related to zoonotic diseases, and then I will know their abundance and their diversity, so it will be easy for decision makers to implement some strategic controls on those areas.
My research is about the prevalence and antimicrobial resistance profile of foodborne Salmonella and E. coli associated with house rodent and shrew in Morogoro municipality. Rodent are reported as a transmitter and the carriers of pathogen, including Salmonella and E. coli. That's why I was interested to know if the rodent can transmit those diseases, Salmonella and E. coli, to humans through food, through environment, through their feces. The finding I will get, it will be added on the existing ones. It will help the decision maker to know how they can manage the diseases from rodent and rodent also.
My research topics is on effect of blood host plasticity on life history traits of tropical bedbug on selected areas in Tanzania. So I was trying to look if I feed them different blood meals and how they impact on their life traits like fecundity, hatchability, survival, and developmental stages. Also, I was looking also on their host-parasite interaction, as I was looking also on the microbial composition and their diversity.
My research was entitled Community Ecology of Rodents in the Afro-Alpine Habitats of Zagwa Salmons Community Conservation Area, which is a locality in Ethiopia. The African rodents in the different biotopes of Africa, their ecology is not well studied. There is a gap in ecological knowledge, so I want to fill that gap and to generate a relatively long-term ecological understanding of the African small mammals.
My PhD work is on ecology of rodents, small mammals, then rodents and shrews in Tigray, northern part of Ethiopia. It's almost in agricultural field. Try to fill the gap on ecology of rodents, especially with those of pest species. And I am trying to look into the seasonal breeding, which is at what time they breed, and the population dynamics, at which time their population will high, and their abundance and their habitat suitability, which means what kind of habitat is suitable for those rodents.